All right. Hello and welcome. Let's start this session. Sorry, it's a little bit late. So we have Mercedeh Movassagh, and she will be talking about mbQTL, Microbial QTL package. So Mercedeh, the stage is yours.
Thank you so much. I just want to say I'm super excited to be here. I was actually supposed to attend in person, but unfortunately, or fortunately, I don't know. I mean, I had to teach in Vietnam, so I couldn't be there in person, but I really wanted to go over the new package we have. So yes, our package is called mbQTL. It's a package for identifying microbial quantitative trait loci and estimation of any sort of SNP and bacterial or other pathogen relationships across patients. Problem is I'm trying to go to the next page, but I can't. Let's see how to do that. Do you guys have to share my slides, or I don't know how it's supposed to be done?
We see your slides.
No, I just can't go forward on the slides.
They should be on your computer and they should be advanced.
Yeah. It's not going forward, but let me stop share and I'll reshare again.
Try again, yes. Let's see.
Oh, there we go. Do you see now? Is it moving?
Yes.
Okay. I can't have you as a tab. That's why. Okay. So I can't see your questions. So if anybody has questions, please speak up or let me know, because I can't see if you've had questions in my screen or not. So what is the big question that this package is asking? It's asking, does a presence of a single nucleotide polymorphism or several of these associated with presence of microbes or other pathogens? It can also show if there's communal relationships between microbes and SNPs. And so what I'm going to do today is basically give you some slides about the overall structure of the package. And then I'm just going to go over the vignette with you guys. And if there's any questions, please let me know. So when we ask, is there an association between SNPs and microbes or SNPs and other pathogens, what we're trying to do is integrate various sequencing data. For example, in this case, we're either going to try to integrate the results of a GWAS analysis or whole genome sequencing with 16S rRNA sequencing or RNA sequencing data that basically has identified pathogens or microbes. And so we're asking again, what are the variants that are associated with the presence of these pathogens? Generally, GWAS data and any sort of like whole genome sequencing analysis, for the most part, assumes normal distribution. However, if you think about 16S rRNA sequencing, best way to present that is using log normal distributions or zero inflated models. And for pathogens that are identified using RNA-seq, they're very zero inflated. So we're really dealing with two types of data. And what we have to do is to come up with the best way to integrate one type to another. Overall, because we're doing predictions on SNPs, we're going to assume normal distributions in a model. However, we're going to get our samples, our pathogen data in a way that could be incorporated and integrated into a GWAS analysis. So what is the overall structure for input of mbQTL? So for 16S rRNA sequencing data and RNA sequencing data, you have abundance file. That could be normalized abundance. As I said, you have to make sure that your data is normalized in a way that you can actually integrate it into a GWAS. Or it could be count, depending on what model you're going to use, and I'll speak a little bit more about that. so I'm going to go back one more time. So this is just the result of a 16S file. Basically, you're dealing with, when I say gene ID, this is basically the genus of your microbes. And I'll show you the dimensions when I go over the vignette. And then you have your samples up here. And then your SNP files would be your SNPs. And then on the call names, it will be your samples. And they will come in 1, 0, or 2. Basically demonstrated if it's like a homozygous, if it's a heterozygous, or homozygous reference or variant per sample for each SNP. And then you also have covariate files, because a lot of times you want to see what is the association of covariates with your models, such as sex, such as disease. And that could actually be, I'm showing right here only qualitative ones, 0 and 1s, but it could also be quantitative. The file formats the package accepts is MR experiments, so an S4 object, or it could accept data frames. And I do that because a lot of times our packages, when S4 objects are used, they're obviously cleaner and better put together. However, a lot of biologists like to use our packages, and when they deal with S4 objects, they get very confused. So I've made it compatible with both. So the overall algorithm uses 3 approaches. One of them is well known, and it's actually used for eQTL analysis. It's the linear regression approach that matrix eQTL uses, and I'll explain that. I basically just built a wrapper around that. The second approach is something we develop. Its basic concept is a correlation method. So what we're doing is trying to understand what is the correlation of taxa, or how best the taxa are related to each other, and how is every taxa associated with a SNP. And then we come up with an estimate correlate. And the higher that estimate of correlation, it means the more there's a relationship. And the third approach is a logistic regression approach, which basically we're assuming a binomial. Is it present or is it not? That requires setting a cutoff. Okay, so quickly on approach 1, I explained what it is, but I just wanted to show you how the files look like, what are the results, and what are the outputs, basically. So you assume your outcome variable is basically the genus or species. Your predictor would be your SNP. And so you're trying to understand what SNP is associated with what genus. And that's what matrix eQTL does. Is pretty good at doing what it does. I did not reinvent any wheel, and I just basically used a wrapper around that. And so I've shown this before. This is your SNP ID. These are your samples. You get heterozygosity information for each sample at each location. And then it looks across and it comes up with an outcome variable when it's doing this, the regression with your 16S data, and also, or your RNA-seq results data. For that, it uses count data. So really not much can be done about that because matrix eQTL just uses count data. And your result would look something like this. So you'll have the SNP, you'll have the genus, and then there's a p-value and an FDR estimated. You get a density plot of the p-values, and then you get basically a QQ plot of expected p-value versus observed p-value. So for the second approach, as I explained before, you're trying to see what is the correlation between your taxa and species. And you can actually identify your taxa and your SNP. And you can actually identify very interesting associations, because what we're trying to see is, are there communities of bacteria that are associated with each other? And within those communities, are they associated with specific SNPs? So this term right here basically shows the strength of the relationship between 2 taxa. And as you can imagine, you're doing this 2 by 2, but there could be a third taxa that is associated with the second taxa, and you can actually see it, and I'll show you how that will look like. And then you're also measuring the goodness of fit of the relationship between your taxa and a SNP. So taxa X with the SNP, and then taxa Y with the SNP. And then you're coming up with a correlation estimate. So here's what basically your output will look like. Again, you have your RSID. You will have a genus, your first genus and your second genus. So assuming, let me show you, obviously the ones that would be the most correlated would be those. There's actually a function that you could remove those. But like, let me show you. Yeah, so for example, this one, you see that Prevotella is pretty associated with Paracagus, and then you have your R square, you have your R value, and you have a estimated row value. And so as a result, if you pull out, which there's a function for that, pull out the most significant associations, you can see, for example, this is pretty high in terms of how correlated they are. Like Pseudomonas and Neisseria seem to have a pretty good correlation with each other. And so with a method like that, you can observe things like this. And heat maps are also one of the things you could do. So it has actually a heat map function, then you could plot it easily. So the colors are basically log of absolute estimated row of taxa X and Y plus 1. So the higher the color, it means the association is the correlation is basically higher. Your x-axis is your SNPs, and your y-axis is your genus. So there's several interesting things to look at here. First off, for example, if you just start from the first row, you can see that there is 3 or 4 bacteria, and these are bacteria genuses. So 3 or 4 bacteria that seem to be pretty much associated with, correlated with this particular SNP. This is one of the things you can observe. And then you have some that are correlated, but not as much. And then some that are just not correlated. But look at these two on chromosome 15. It seems like you can actually see how linkage disequilibrium works. You see these 2 particular SNPs are behaving pretty much, I mean, exactly the same across the genuses. So you can see there's a relationship basically between several microbial communities that are identified in this 16S analysis with 2 SNPs that are pretty close to each other and are in linkage disequilibrium. So this is some of the things that you can find out in terms of, is there a communal relationship between various microbes and the presence of a SNP? And this is something that is pretty light. I mean, if there's a relationship, you suspect that it would be like that, because imagine if there's an immune dysfunction, a mutation related to an immune dysfunction, you would assume communities of bacteria might grow more and more, and the patients would actually show effects on that, show that sensitivity of a microbe relationship with that SNP, not just one. And so for approach C, you're basically assuming a 0-1 situation where you assume, is there a microbe present or absent? And is there a SNP present and absent in terms of, but when you're assuming present and absent, you're also assuming the heterozygosity of the SNP. So this is a scenario that you could be looking at. For example, Neisseria right here is not present. I should show that there could be a situation where there's one, but you'll see that when I actually run the code. I just took some pictures of things. So your output would look like this. You'll have your RSID. I mean, the statistics information is there. Obviously, there's an FDR as well. I'll show you when I run the code. But basically, you'll see with a specific chromosome, is there a specific relationship with a bacteria? For example, these are relationships with Neisseria. And you have to do that one by one, by the way. You could run all of it at the same time, but the relationship would be estimated per bacteria or pathogen. So you get plots like that where you look at specific alleles that could be associated with presence of bacteria or pathogens. For example, in here, you can see that GG is associated highly with presence of Neisseria versus AA, haplotype is not actually associated with the presence. It's the other way around. So most of the hosts that have the AA don't really have Neisseria. And so this is the overall picture of the package. But now I just wanted to show you guys some of how the code is run and how things are being done when we run it. It is pretty fast. I mean, you'll see some of the timings as well. So let's do this. Okay. Did it run? Okay. So yeah, so this is just an example of how the metagenome object looks like. It's just an MR experiment, but just to give you an overview of how these files look like. So yeah, as I explained to you before, you have the samples, you have the pathogens. This is your microbial abundance file. Then you have your SNP file, which is the samples and the SNPs. And then the covariance file, which, as you can see, there's sex and disease. Okay. So the first analysis that we want to do is to do like a linear regression analysis using matrix qTL. And, oh, you can't hear me? Sorry. Hold on. Yeah, I can't hear you guys too for some reason. Okay, yeah.
Hi, Mercedeh. I just thought I'd update you that online we can hear you just fine, but we're just seeing your, and this is probably the same in the room, your slide that says the vignette. We're not seeing your sharing of another run screen.
Okay, I'll stop sharing then and let me see. Maybe this, yeah, it's funny. Let's see. Now I can't even share, it seems. Yeah, I can't share right now.
Okay, Mikhail, I think you're the only moderator here. I don't have moderator privileges on this one, so I can't.
Okay, now I can. Hold on.
Okay, great.
Let's see. Share. There you go. Do you see this?
Yes, looks perfect
Okay.
now. Now we see
Cool.
your RStudio.
I see. But I did also, so Dave told me the last part they couldn't hear, part C. Is that correct?
We heard you online, but I guess, yeah, the
Okay.
room lost its audio for a minute.
Okay, so I guess I have to stop sharing, go to the tab, and then I have to switch and sharing.
Or just share your whole screen so that you can go back and forth, I guess.
I am going, so I think the problem is if it's a tab, it would do the actual presentation, but if I want to do the RStudio sharing, I have to actually go to a window. That's
Right.
the problem. It's probably because I don't have a screen, an additional screen.
Okay. I'll stay on hand here and I'll let you know if we're not seeing something.
Perfect.
All right.
Thanks. Okay. So I just want to make sure you all are seeing this. Is it good? Can someone just tell me yes in the audience in the room? Yes. Okay, yes. Cool. Very good. Let me go back to this then. Okay, so I think you stopped hearing me after we got to this section, which was the logistic regression section. For this, we're trying to basically understand if presence or absence of a microbe would be associated in like a 0-1 format, in a binary format, will be associated with specific SNPs or not. And so in this particular example, we're looking at Neisseria. You could do this across all of the data set, but for this, and I'll show you, there's actually a function for that. But for this, I just looked at I'm showing you 1-to-1 relationship. Actually, somebody, okay, cool. So yeah, so as you can see, so what we're doing is just simple logistic regression. We estimate p-values and FDR for this. And as an example, you could see this particular SNP on chromosome 2 seems to be significantly associated with Neisseria bacteria presence or absence. In particular, if you have this haplotype, GG, it seems like it is associated with 1 shows presence of a Neisseria, versus if you have AA allele, you won't have association with Neisseria. It's actually higher with 0, which means Neisseria is not present. Okay, here's where the vignette is. Now I'm just going to switch my screen and go to RStudios to go over the vignette with you guys. I'm going to stop sharing real quick and then share again. Oh, now it's not allowing me actually to. Hold on. There you go. Oh, cool. Okay, so yeah, let's load some libraries and also some of the data, and I'll show you how the data looks like. So, for example, the microbial abundance file. As I explained before, your row names is your sample names, and your col names is your bacteria or pathogen. Your SNP file is your SNPs. SNPs again on the col names, and on the row names you have the sample names, and your covariate file has a covariates the other way around. So now your col names would be your samples and your row names would be your covariates. And then this is just the MR experiment object. That is a metagenome-seq object. I'll explain to you as I go down, but you don't have to stick with metagenome-seq. You can use phyloseq as well. I have functions that are adaptable to that if you don't want to use log normal distributions. So here's what I was talking about. So if you want to have a compatible, try to make metagenome-seq object compatible with this package, we've built a function in there. It's called metagenome-seq to mbQTL. At the same, and this does log normal distribution. It basically normalizes your data using log normal distribution. However, if you want to use phyloseq to do your own sort of analysis, phyloseq actually has a function that's compatible with metagenome-seq, and you could just do, it's called phyloseq to metagenome-seq. You could just change it to that, and from there get it and change it to this. And if you're using a data frame, you don't need to change anything. So now the first step is to do the linear regression. Again, what we're trying to do here is to understand if specific SNPs are associated with specific taxa. We're using matrix QTL for that. And it is pretty fast. As you can see, it's like extremely fast, and you can get your results quite fast. And you get, here's your SNPs, your genus, p-value, and FDR, if there's some sort of association. Keep in mind that it is mbQTLs are just very hard to get to specific significance. So a lot of times, unless your data is like completely like associated, it's very hard to get a very significant FDR out of this data. And so you can draw histograms for the p-values. You can create QQ plots, where's the expected versus observed Q. It jumped up, sorry. Whereas expected versus observed p-value. And well, you could go to the video and look at this, but I can't really quite make it bigger. And then you could also do the correlation. So for correlation, I made it so that it's step by step, because it might be that some people are interested in having these values. So this particular one gives you the R value, which is the correlation value between your two genuses of either bacteria and bacteria, or like virus and virus. You can have this value as well. Then you could look at what is the relationship between the RSIDs and genus 1 multiplied by RSID by genus 2, so product of R square. And the next step is trying to understand what is the estimated row value, basically the product of row. Let me see. Okay. Yeah. So this is the actual estimated row. And so you can plot the heat map for this, and you could set like cutoffs for how much of an association you want to see. Like you want to see the top 99% of the association or the top 50%. So that's probs is how you set it for. And then you can plot it. And this is something like I just showed you. You have your organisms, pathogens, and you have your SNPs, and you can see the relationships between them. And so the final step is the logistic regression. This one takes a little bit more time, but overall, like, and I have to show you how big the data set is, but I'll explain that after the vignette's done. Yeah, so you could look at a relationship of taxa with SNPs with various microbes in this case, or pathogens. And we can we calculate p-value and FDR. We also do Bonferroni correction. Yeah, I have just two of this. So this was with all of the, this one was with all of the microbes. This one is just one-to-one. Is there a relationship between specific SNP and a specific microbe, not all of the microbes. So basically our corrections and p-value, FDR corrections would be just based on one, not all. That would be the difference. And you can plot this, easily plot it with the legit plot SNP taxa. So again, pathogen absence, pathogen presence, you could name whatever you want, but this is, we are asking for this particular SNP, but you could put any SNP that you want, basically, as long as it matches your SNP name. And then it would show the alleles and what is the difference between presence and absence of that particular pathogen. So yeah, so this is basically overall, this is pretty fast. Most of it, depending on what the size is, you could really run in like 5 minutes, the whole analysis. However, it depends on the size of the files. This particular analysis was with around 1,500 genus of microbes. And then it was also, I think, 20, because it was a result of a GWAS, 20 SNPs. As you can imagine, as you increase the number of your SNPs, so if you were to put your whole genome sequencing results in there, it would take more. But I've done like around 1,000 SNPs with around 500, 600 taxa. And the longest was a logistic regression that took like 3 minutes to run it. Yeah, other than that, it was pretty fast. Okay, so this is overall what we have. If there's questions about it, please let me know. Sorry. You mentioned that microbial data are zero inflated. Did you consider a Poisson or negative binomial models for the QTL mapping? So the QTL mapping, actually the actual mapping assumes normalized data, because it's GWAS analysis, and most of it is assumption of normalized data. Normal distribution. It's too late here. It's like 11 at night, so sorry about that. It assumes normal distribution. However, when you're doing normalization before you input your 16S analysis, you actually do log normal distribution. So that's why you have that auxiliary function that's like metagenome seq to mbQTL, because you're trying to normalize your data. Or if you're trying to do your own normalization using any other method, you could do it and just incorporate it in there. But the overall, so two of the methods assume just normal distribution. The last one is a binomial assumption. Okay, any other question? Or was that question answered fully? And I did not assume negative binomial, but you can assume that, and then you can input that data in there.
I had a question. Have you had a good experience with metagenome seq? I've had some concerns about its false positives.
So generally, 16S data has a lot of false positives dealing with it. I guess, so I haven't developed metagenome seq, to be honest, so I can't really answer for that. It does model it pretty well. I personally just do normalization the simple way, basically. But a lot of people I know have had good experience with it, so they use that. That's why I made it applicable. There's a lot of zero inflation in there too. So I've tried zero inflated models. They were fine. Zero inflated negative binomials were pretty good. So Zimb was a package I used, and it was pretty good. Zimbwave, yeah.
Zimbwave.
Yeah.
Great. Well, I wasn't planning on being like a moderator or chair of this session, but it kind of seems like we've lost the room. And so we're just the online audience here, as far as I can see.
So I think there's another question Jill is asking. Are those p-values? Okay. Are those for correlations, the regression coefficients? So the p-values, every section has, I might have gone over, I'm pretty much sure I went over it really fast, but for all of them, you generate p-values or regressions. So every step, basically. So if you're doing the matrix QTL step, the first step, approach A, it will actually generate p-values for what is the basically expected p-value in terms of association of taxa and SNP. across the data set using a linear regression. And then it also does FDR correction. For part 2, you're just looking at correlation. We don't do p-values because we're looking at what is the strength of this. So you do probabilities. You say, okay, I want the top 1% of the highest correlation. So you just show that. Obviously, I thought about actually assuming a background distribution and setting up p-values that way. And then the third part, yes, you can just do a logistic regression, and from that you get a p-value and you do an FDR correction based on the number of p-values that you get. I hope that answered that. Let's answer many. Function is used. Much better. Yeah, so fit feature model, that's true. Yes, that's exact. So Mathilde was just saying use a fit feature model is better and it controls the false positive. Yes, that I actually, when I used it, I saw that as well. That has less false positives. Cool, so Jill is fine. I hope I did not miss anyone's question. Okay. So anything else? So another thing that I want to say is that, so approach A is generally what is used in publications separately, but people are just adapting basically. Approach B has never been used, and approach C is a lot of publications use it, but it's a very, people use it in a messy way. So I guess one of the most important reasons that we developed this package was because we wanted the community to use the same thing. So so if there's problems, there's reproducibility issues always. So we can go back and fix it. So everybody uses the same package, not just there's a piece of code written by someone and we're gonna be okay, great, you're right. But that was one of the reasons we wanted to write the package, actually. Okay. Any other questions? I think. Now you're on mute, Levy.
I just wanted to add that I'm really happy that you have published this package. It's a mess previously.
It is a mess.
Kinds of analyses.
Yeah, it is a mess, and I think everybody just uses this a different way, and it can't be controlled. That was the biggest concern. I think actually it works pretty well because so as you pointed out, generally 16S data is very noisy and messy. But I think another thing to point out is that RNA-seq is now doing really well with pathogen identification and also microbes. Because before you can really do microbes with RNA-seq, but now Chan Zuckerberg has CZID, which they're doing pretty well at detecting bacteria and such. So the noise has become less. So if you standardize the downstream analysis, I think it's going to be very good. It has a bright future, I think. Okay. So yeah, I mean, if you guys had any questions, yeah, I mean, send me an email. I'm happy to talk, collaborate, and improve this. This is just the beginning. I've just started doing some microbiology, as I should say. So that's why I developed this. But I'm sure there's a lot to be improved in this. This is very preliminary, I think.
In case anyone hasn't joined yet, there's a hashtag BioC 2023 channel in the Community BioC Slack team. So
they lost the audio again.
It's a good place to continue the conversation.
Yeah.
So far, it's been kind of the story that that we have more glitches for the online audience with an in-person speaker and more glitches for the in-person audience when we have an online speaker. It's been perfect for the online audience.
Yeah. So it's good that I'm virtual. I'll listen to all the talks with no problem. Good. Yeah. Okay, well, I really hope that you guys contact me if you have better ideas and we can develop this further, because I think there's a lot of space for developing this. These were just simple models that we applied. Yep.
It's fantastic.
Thank you so much.
Thank you for joining us in the middle of the night.
Of course. It's so exciting, and I'm so sad that I missed Biocy this year.
Me too.
Okay. Okay. Well, all here talks virtually, and thank you again for the invite. Okay. Have a lovely night.